Swayam Prabha. Digital India. Educated India.
Welcome you all to the next class on Inorganic Chemistry of Life, Principles and Perspectives. Immediately prior to this particular lecture, we had completed the iron over a period of 10 lectures or so, all different types of enzymes, storage, transport, as well as the oxidative type, reductive type, hydrolytic type. More or less a good range has been covered. So, let us now get into the next one in the periodic table. After the iron, what comes is the cobalt. Unlike the iron, cobalt is involved in only one particular case, and that is vitamin B12. We all know that vitamin B12. Actually, vitamin B12 is just a coenzyme, it is not an enzyme. And therefore, a coenzyme containing adenosyl cobalamin acts as a coenzyme with a host of enzymes and does a lot of reactions. So, therefore, you can see that water soluble vitamins has a key role in brain and nervous systems and functions very well. And it is one of the 8 B vitamins. It is involved in a variety of metabolic processes, particularly DNA synthesis, regulation, catabolism of fatty acids, some amino acids like threonine, methionine, and branched ones like leucine, isoleucine, variety of these things involved. But only one enzyme has got the cobalt in the entire thing, whereas iron is present in more than 200, 300, 400 kind of different enzymes, that you see the difference. Also, cobalt is not present directly in the enzyme, it is present in the form of adenosyl cobalamin, which is associated with a huge host of enzymes and does the function. How it does, we will learn in this particular class. Okay. So, it is one of the rarest one that we have for the cobalt, and that is what we understand that. Okay, and it is produced by the anaerobic bacteria like Streptomyces species, and not by the plants and animals. So, this is also having anti-pernicious anemia factor used for the treatment of pernicious anemia. So, people use, you know very well, vitamin B12 they take for whenever you feel somewhat weak, you take that one. That is how you supplement the cobalt in the form of a cobalamin and some kind of a supplements in that too. Okay, now let us look at some of these different forms. This we can build in the forms. It has see that this looks like a porphyrin ring, but if you look at close by, it is not, because there are some saturated centers there, and this is known as a Corin ring. This is known as Corin ring, as you can see that. Not heme, not porphyrin. It is somewhat like reduced porphyrin or something, you can say. Now, so that is bound through the 4 nitrogens over here, and of course, it has its own. And then the one more group is possible, and this is, what is this? You can see that this is connected through this, and if you look at here, this is the benzimidazole, and this is the glyco kind of a moiety and the phosphate. So, this is more or less like a nucleotide. So, it is bound through a nitrogen of the nucleotide. Okay. And then this side you have the some groups depending upon during the different kinds of enzymes. Most of the enzymes will work with adenosyl, but some enzymes will work with methyl, and some enzymes, some during the isolation process can cyanide, other kinds of things are also hydroxy, all these things can form. Okay. So, it has a nucleotide, it has a tetrapyrrole ring which is corin, it has a cobalt of course at the center and it is an R group. If the R group is cyanide, then you call it cyanocobalamin. If an R group is a hydroxy, then you call it a hydroxycobalamin. Of course, you can say aquacobalamin. And when you have a adenosyl moiety, which I will show in the later slides, then adenosyl cobalamin, and if it is a methylcobalamin. So, I will show some examples. Most of the examples are by adenosyl cobalamin, and one example I will show by the methylcobalamin too, how they are all involved along with the corresponding enzyme and function. So, this is whole thing is called the cobalt based cobalamine coenzyme. Primarily when R is adenosyl amine, adenosyl, then it is called adenosyl cobalamin, that is the one which is involved. In some cases methyl, some cases cyano, etc. In different forms it will change. So, we have looked at this, then we can also look at the cobalt center. The cobalt can be in the cobalt 3 form, cobalt can be in the cobalt 2 form, cobalt can be in the cobalt 1 form. When it is in 3, then it is referred as a normal, then when it is 2 is reduced, when it is 1 super reduced. How will it happen? It depends upon what kind of a binding, how that changes. It depends on the binding and the X group both. It depends on both the binding. So, the binding in this case is through the that nucleotide, benzimidazole based nucleotide, as I showed on the previous slide, and X could be various things. So, this particular thing is basically referred as the adenosyl. So, okay, so that is adenosyl cobalamine when it is bound. So, whether the cobalt is 3, 2 or 1 also depend on the X and its bulkiness, etc. I will explain in the thing. When you have water, it is called B12 echo. When you have hydroxo, it is called hydroxycobalamin. This is B12A, B12B, and B12R is cobalt 2, B12S is super reduced cobalt 1. B12 is cobalt 3. So, when you write B12 is cobalt 3, and this bond can change depending upon the X that you have, and you can see this, the cobalt 3 is with a perfect normal binding with a 6 coordinate, the cobalt 2 with a 5 coordinate, one of these is basically broken kind of thing. Cobalt 1, both of them are broken because very strongly electron dense kind of a center binds that. So, 6 coordinated, 5 coordinated and 4 coordinated. So, from that also one can try to identify. This is a synthetic kind of thing, where X and Y can be varied, and then people have made all these kinds of, these are called cobalt oximes. So, like other kinds of oximes, cobalt oxime. So, let us come to the situation of the cobalt carbon bond. As I told you earlier that the cobalt can be in 3, 2 and 1, and so cobalt R bond, if it is cobalt 3, the R will get a characteristic of R minus, okay, and if the cobalt is cobalt 2 characteristic, then the R will be like R dot, and the cobalt is cobalt 1, then R will be R plus. So, in this case it is the cobalt 3, cobalt 2, cobalt 1, R minus, R dot, R plus. What does that depend on? First of all, what is a cobalt carbon bond? Cobalt carbon bond in these things requires energy of about 25 to 30 kilocalories, which is generally higher than for the catalytic kind of thing. So, therefore, one needs certain kind of a rearrangement, which is also dependent on the X group that you have. So, the one is axial ligand, and the bond dissociation energies are accompanied by the reduction of the cobalt center from cobalt 2 to cobalt 1, and decrease in the electron. If you decrease the electron donating ability of this fifth coordination or the axial ligand, then you can stabilize the cobalt 2. If more electron donating property, it will become cobalt 1 and R plus. If it is much lower in electron donating then it will be cobalt 3 and R minus. So, these are the things. So, therefore, all these will, and the second important very point is the X group its steric parameter. So, because this whole thing is going to interact with the enzyme. So, therefore, the R group that is present, X group that is present on the cobalt center between the chlorine ring and this one. So, in the case of adenosyl cobalamine, it influences the strength of the cobalt C bond. So, whatever you have in the 6th coordination, its steric bulkiness, its interaction with the, when it interacts with the enzyme, how the conformational changes will influence the bond, these all will decide the breakage. So, essentially whether it will break as cobalt 3 R plus, cobalt 2 R dot, cobalt 1 R minus, cobalt 1 R plus, sorry, cobalt 3 R minus, cobalt 2 R dot, cobalt 1 R plus. So, these are the kinds of things. So, having looked at that, let us try to understand this different electronic states. When you have a cobalt 3, okay, you have a 6 electrons and the cobalt 2, the 7 electrons D, and cobalt 1 8 electrons. So, if you look at this and try to fill this, and they are all filled early, and there is a empty orbitals and the ones which you have these empty orbitals. That means it is capable, it is like a Lewis acid. So, it is capable of accepting an electron pair low lying on the empty orbital. Now, you come to this, the 7th electron is here, therefore, this electron only one single, therefore, it is a radical kind of thing. So, radicals can be very reactive too. And if you look at this one, you have both the electrons are filled. So, it is a pair. So, there will be a nucleophile. So, electrophile, radical, nucleophile. This is B12, this is B12R, B12S. So, these are the. So, B12 is cobalt 3, B12R is cobalt 2, B12S is cobalt 1, and that is what we get. Now, having looked at the characteristics of the cobalt center oxidation state, binding from the benzimidazole, binding from the X point of view, and whole thing interacting with the protein, and what kind of reactions these guys are involved, the cobalamine based enzymes. Basically, they do a kind of a breakage and makeage, break a bond and make a bond. So, you can see this example C1, C2, the C1 has X, and on this side C2 has X. So, it is because there is a hydrogen and shift, it is called a hydride shift, in this case 1,2 hydride shift, 1,2 hydride shift. And if the X is acyl or alkyl group, it is called class A kind of a reaction, and this is again different enzymes are associated. And if you have a nitrogen containing ligand, it will be class B. If you have OH or amine, and at the end the water is eliminated or ammonia is eliminated, then such a kind of class of reactions are class C. No elimination in A and B, there is a water or ammonia elimination in class C. So, overall the cobalamine enzymes, cobalamine coenzymes are associated with a host of enzymes, and does the reaction of A type, class A type, does the reactions of class B type, does the reactions of class C type, okay, and that is clear. So, what would happen at the adenosyl center? That is, let us look at. This is a part of adenosyl and the cobalt carbon bond. So, through this is not from the other side, this is from the adenosyl side, and this adjusts in such a way, because its bulkiness in such a way, it always goes to the cobalt 2 with this particular radical here. Actually, CH2 is a radical, and such a radical can induce a radical in the substrate and transfer the hydrogen to this and become CH3. So, the CH2 radical can become CH3, and then radical is associated here, and then that radical can rearrange, etc., etc. So, you generate a radical by breaking the cobalt carbon bond at the cobalt adenosyl, and this radical is transferred to the substrate, and substrate gives the hydride and becomes methyl, and again goes to substrate radical, and substrate can make any kind of a changes and become a product. And now, this is transformed to methyl, then there is another set of enzyme which will remove this hydrogen as a H dot and then go back to the cycle kind of thing, Okay. So, this whole thing occurs at an interface of the vitamin B12, which is a coenzyme, and the corresponding enzyme. So, is that understand? We have explained what are the different kinds of things, then we have looked at the what different types of cobalts 1, 2, 3, and what kind of an X group and adenosyl, how does that break. So, I said the reactions of the A type, reactions of the B type, reactions of the C type. Let us look at some of the reactions. For example, glutamine mutase. If you look at this and one of these carbon, there is a shift, 1,2 hydride shift, that becomes methyl group. So, this goes and connects here and this becomes CH3, and that is where the 1,2 hydride shift. Here you have this methyl that will get converted into methylene and then get inserted into this. So, it is a 1,2 hydride shift. Again here another 1,2 hydride shift. So, these are all A type kind of things where you have the groups of the mentioned there. So, these are mutases, mutase. So, all these mutases obviously are involved in 1,2 hydride shift. Then B type where nitrogen kind of ligands are there, but no elimination takes place, and you see that, and this shifts over here. So, NH2 and methyl group, and shifts over here. So, NH2 and methyl group. So, this is lysine mutase, alpha lysine mutase, beta lysine mutase. It is NH2 group, nitrogen containing, but no elimination of anything happens. So, class A type 1,2 hydride shift, class B type hydride shift, but having a nitrogen. So, that will become amine group and that will rearrange. And then you have a class 3 type, class C type, where it is after the 1,2 hydride shift, there is an elimination of water or ammonia. Look at the glycerol dehydrase, and this one, this will become the 1,2 shift, that will go into 2 hydroxyl groups on the same, which is called gem dihydroxy, and this is, we know very well from the organic chemistry, this kind of a situation most often leads to the elimination of water and then gives this one obviously. And another example, propane 1,2 diol dehydratase, again will go to the aldehyde. So, this kind of reactions are type C, and in this case it is the amine which you have. So, therefore, it is the NH2 plus H, that is ammonia is lost, and then so either the ammonia loss or the water loss, any of these things will happen with this too. These are class 3. So, now I explained you the class A type of reactions, class B type of reactions, class C type of reactions in all these things, and let us look at their mechanistic aspects. Let us look at the last example. We have taken diol dehydratase. Diol dehydratase means water kind of thing. So, the enzyme, let us take in the normal situation cobalamine adenosyl. This is the adenosyl cobalt, this is the rest of the enzyme. So, this enzyme cobalamine enzyme, these are associated, this is coenzyme. Now, when the substrate comes in contact, obviously substrate has to bind in very close proximity of this the adenosyl group, because the radical has to be transferred without any other destroying or other kinds of things happening. So, therefore, it will bind very close proximity of the cobalt center, cobalt carbon bond center from the enzyme side. When it binds, there is certain kind of a the conformational change that occurs that will trigger the cobalt CH2 bond to weaken this bond. So, when it is weakened, then this will hydrogen abstraction of H, and that will lead to the this kind of things. You see that, and starts breaking, and then you get the radical part, and this radical obviously has gone to this. So, the rupturing hydride, and therefore, it becomes the radical, and this radical is transformed or racemized to the next carbon, that is also same as saying that hydroxyl is shifted to this or migrated to that. So, that is again hydride shift, 1,2 hydride shift. So, now you find a situation where this is all on the substrate, and that substrate is binding very close to this. You see that, very close to that. That brings a lot of conformational change, and that conformational change is the one which is responsible for weakening this bond and make the radical, and the radical abstracts the hydrogen or hydride, hydrogen dot, and give the radical on the substrate radical. This is a substrate radical, the radical shift happening because of the hydride shift. Now, this particular thing is not stable, and this will go by removing the OH and H as a water. So, when you remove the water, you get this, obviously the dice. Okay? You can see that, and now this one again has to bind because it is the CH3 has to make the form of the binding should take place normal. So, therefore, cycle is regenerated. So, you understand the cobalt adenosyl bond breakage, and then the radical, that radical is transferred to the substrate, and the hydride is transferred to the adenosyl to become CH3, and then that radical is racemized, and then in that process you have a hydroxyl shift, and then 2 hydroxyl will come on the same carbon and loses the water, and then makes this one. Okay. Now, not only the adenosyl, even the methylcobalamin in the also involved in homocysteine to methionine. Okay. First, you have a biologically important thing is not methyl one, it is adenosyl one. Adenosyl one will lose this one and get the methyl group from this particular moiety. This is called N5-methyl tetrahydrofolate. From here the methyl comes here, as you can see the methyl, of course, in an inverted fashion, and this is the one which is interacting with the homocysteine, and the homocysteine this gets inserted, and therefore, you have the whole thing going into this. So, I have not shown all the other parts. So, it will convert into the methionine. So, homocysteine to methionine. So, cobalamin is obviously required for the conversion of homocysteine to methionine, and it is not the normal cobalamin, it is the methylcobalamin. So, the normal cobalamin is adenosyl cobalamin is loses the adenosyl and methylation takes place. Cobalamin must first undergo the methyl transfer to form methylcobalamin. So, it receives the methyl group from N5-methyl tetrahydrofolate, thus regenerating the tetrahydrofolate to participate in other 1 carbon transfers in purine metabolism or pyrimidine kind of a thing. So, these are all kind of things that happen, that is why DNA synthesis and other things are there. Now, so, we have seen one diol dehydratase, then we have looked at the homocysteine to this methionine conversion, and for which you require the methylcobalamin. And now, we have another example, ribonucleotide reductase. We have already seen under the iron what a ribonucleotide reductase is. Ribonucleotide reductase is the enzyme which converts the ribose into the deoxyribose, but the entry point is the nucleotide of the ribosyl and then goes into the deoxyribosyl. Okay. So, this I have fully explained to you under the ribonucleotide reductase under the iron completely. As I told you that there is the center the diion center the ion with the tyrosine radical. This radical is transferred to the second portion of this particular enzyme through the cysteine to the to the center of the ribosyl to the C3 prime, then converts to C2 prime, and then finally hydratase kind of water, then giving that we have seen already. So, C2OH and C2H, and this is the ribonucleotide reductase activity, and this is also done by adenosyl cobalamin based enzyme. And you see, enzyme is very complicated, you have so many lobes etc. are involved in all of these, and then you can see that yeah. So, how does this happen? Happen very similar to what I have mentioned in the previous case, where the the diol dehydratase in some context. Now, let us look at in this enzyme. Again, you have a coenzyme, vitamin B12 coenzyme. In the enzyme, you have something more very close, where it reacts, where it interacts with the cobalt this enzyme, there is a sulfhydryl functions, dithio functions are involved. The protein based dithio functions are involved. So, therefore, it is somewhat analogous to that what we talked in case of the iron, though in the case of the iron the radical is transferred via the cysteine, via the glycome via the ribosyl moiety to the dithiol function, but here it is very close proximity. Okay. When this particular substrate binds to the enzyme, that is nucleotide binds to the enzyme, this cobalt adenosyl part will get influenced, get strained, and then can give the breakdown of the cobalt and carbon bond to give a radical. And this radical now earlier, in the earlier case it was directly transferred to the substrate which is diol, but here it is not transferred to the substrate, it is transferred to the enzyme containing dithiol moiety. So, therefore, you have the cobalt 2 and the carbon center, this radical is transferred to the dithiol of the actual protein, and that in turn is transferred to the ribosyl part of the substrate. So, this is not from the adenosyl, this is from the substrate. So, in the substrate from 3 prime position, this is the 2 prime position, and this the 2 prime position you will lose the in presence of the proton it will lose the water, and then you have a hydride, and that leads to the kind of a thing that you have enzyme with OH and the S, and that results the returning of the cobalt carbon bond in this, and this leads to an oxidized form. And there are other enzymes which will again reduce this, then the enzyme is available for catalysis. Okay. So, therefore, in this case you have the cobalamin adenosyl cobalamin coenzyme, and you have an enzyme portion at the interface, you have a dithiol function which is in close proximity. When the substrate here is a ribonucleotide, when it binds, then there is a change in the conformation that will make the cobalt carbon bond somewhat weaker, and that can break down to the cobalt carbon as a radical which is transferred to the dithiol function and to that of the substrate in the ribosyl part, and then the ribosyl part accordingly loses its particular the OH with the addition of the H as water, and then that dithiol moiety undergoes oxidation, and this oxidation will bring down back to this particular thing. And now, if the enzyme has to function, this disulfide kind of a formation or oxidized dithiol has to be re-reduced back to the this, then is ready for the next cycle of this one. Okay. So, in effect, what we have learned unlike iron, cobalt is involved in only one the coenzyme, that is called vitamin B12, where the adenosyl is involved, whereas iron is involved in hundreds of enzymes and variety of reactions. And in case of the cobalamin, all the reactions are of the hydride shift kind of a thing, and in most of these cases is the cobalt and carbon bond is broken in the form of a cobalt 2 and the carbon dot. For this what is required is the binding of the substrate, and the adenosyl moiety itself provides a steric strain, and you know this vitamin B12 coordinates or combines, not coordinates, combines with that of the main enzyme and the substrate binding. These will bring a lot of change in the steric factors at the center of the cobalt carbon bond of the adenosyl, and that will weaken the bond, and that bond will rupture or break, and that is one which is resulting in the form of a radical. And then the rest of the story is either the radical is transferred to the substrate or the radical is transferred to the thiol moiety in one case, and then transferred to the substrate part of it. So, we have 3 different classes of reactions, class A, class B, class C, but in all the cases it is the hydride shift which is important. In the class A you have acyl groups and other ones, class B you have nitrogen containing groups, but in the transformation no breakage or no loss of any moiety occurs. In class C, the transformation of the 1,2 hydride shift will transform as a hydroxyl group or amine group, and in case of amine group it will be ammonia which is eliminated. In case of the OH it is the water that is eliminated, because germ di hydroxy groups are not stable and they will lose. So, overall what we say is that the cobalt carbon bond is broken in the form of cobalt 2 carbon dot, particularly in case of adenosyl cobalamine, and then the reactions go through the radical kind of a mechanism. So, this is only one enzyme. So, therefore, I will stop at this stage for the cobalt, and in the next class I will take up the nickel containing enzymes, which I will continue for 4-5 lectures or so. Okay. Thank you very much.